Advanced cognition depends on the dynamic coordination of information flows. While neurons provide the hardware, the Brain Kinematics Model suggests that intelligence is found in the fluid routing of these signals through shifting pathways. If raw brain volume or total neuron count directly dictated behavioral flexibility, the largest animals would possess the highest reasoning capabilities. A sperm whale's brain weighs nearly 8,000 grams, yet its cognitive output remains specialized rather than abstract. Look at this photograph of a raven. Corvids exhibit highly complex cognition, including multi-step problem-solving and tool use, despite operating with a brain the size of a walnut. Their cognitive power is a product of efficient routing and dense organization. Evolutionary advancement layers meta-control architectures over existing reflexive modules. These higher-order systems suppress, recombine, and reroute hardwired circuits, allowing the organism to adapt to environments the original circuits were never designed for. To understand advanced cognition, we must look past structural anatomy and analyze the physics of signal routing, a metric we call degrees of freedom, representing the number of distinct behavioral pathways available to an organism. This structural problem is addressed by the Brain Kinematics Model, or BKM. It is a unified framework designed to decode neural evolution and resolve the paradox of brain size versus cognitive capability. The core premise of the BKM is that neural activity does not operate like static electrical circuits on a motherboard. Information behaves as structured, fluid pathways known as strain flows. These strain flows are constantly shaped, bent, and routed by two physical forces: the precise delays in the brain's wiring and hierarchical attractors, which are specialized regions that pull data toward them like gravitational fields. Under this model, a behavior is never a fixed file retrieved from a hard drive. It is a temporal pathway, continuously competing against other pathways for physical dominance within the brain's real-time environment. In the BKM framework, the foundational biological unit is the module. A module is a local strain package, a highly specialized microcircuit performing a single function, like detecting a visual edge or a sudden pulse of fear. Evolutionary scaling begins when these isolated, rigid modules couple together. They form functional groups that fire as a cohesive activity block, allowing a sensory module to link directly with a motor module to produce a coordinated response. Integration scales up again by coordinating multiple groups across long distances in the brain. This creates a distributed network, physically connecting perception, memory, and action into a single complex task. Because these connections are dynamic, the brain relies heavily on reusability. A single local module, such as an edge detector for hunting prey, can be dynamically recruited into an entirely different network for navigating a physical maze. Evolutionary complexity scales vertically through this dynamic recombination and coupling. The brain gains capability by integrating existing strain packages in increasingly sophisticated ways, rather than building isolated new circuits from scratch. There is a common assumption in biology that evolutionary progression strictly favors maximum signal conduction velocity, the idea that a faster transmission always yields a smarter brain. Uniform maximum velocity across a nervous system is physically dangerous. If every signal traveled at top speed, regardless of the physical distance it needed to cross, the result would be chaotic signal mismatch and a total loss of network synchronization. This graph plots axon conduction speed against phase coherence. Peak efficiency occurs far before maximum velocity. Evolution optimized delay distributions, varying axon diameter and myelin thickness to strictly control timing, rather than maximizing raw speed. By tuning these delay distributions, signals traveling from vastly different distances can arrive at a target node at the exact same millisecond. When this perfect phase alignment occurs repeatedly across multiple oscillatory cycles, it creates phase waves. These are emergent, traveling waves of activity rippling through the cortical tissue in perfect time. As these cascading waves lock together, they form the BKM continuum. This is a stable, large-scale, phase-locked neural field, the physical substrate necessary to hold a thought or a perception in place. Higher cognition is biophysically impossible without these specifically tuned delay distributions. They are the structural mechanism holding the temporal continuum together. A massive, synchronized cortical continuum is computationally powerful, but without directed top-down control, it leads to behavioral paralysis. The brain must choose one coherent action out of thousands of conflicting possibilities. This selection is managed by the BKM attractor hierarchy. Specific brain regions act as physical gravitational forces, routing and prioritizing the neural strain flows to force a decision. Later one is the amygdala. It functions as a polarity attractor, rapidly biasing the entire network by tagging specific sensory fragments with emotional urgency, elevating the priority of a threat or a reward. Layer 2 is the hippocampus. Operating as a longitudinal attractor, it indexes and binds dispersed cortical packages together. It creates continuous spatial routes, or episodic memories, structuring data into sequences known as mesothemes. Layer 3 is the prefrontal cortex, or PFC. This serves as the high-order attractor. It is the executive selector that suppresses competing pathways and organizes recursive, self-referential loops for complex future planning. Abstract, conscious thought emerges strictly when these three nested gravitational fields successfully organize and override lower-level sensory flows, pulling the system into a unified behavioral output. We can formalize this hierarchy into a competitive mathematical space, calculating exactly how a specific neural pathway wins dominance using a differential equation for pathway activation. This equation calculates the strength of a neural pathway over time. Activation relies on baseline structural inputs, the direct sensory match, alpha II, plus the emotional amplitude from the amygdala, beta AI. These act as physical pressures widening the strain channel. The pathway also requires high-order structural inputs. The gamma HI term applies contextual support from the hippocampus, while the delta PI term applies top-down executive weighting from the PFC. To survive, the pathway must overcome opposing resistive forces. The negative terms in the equation represent the brain's baseline passive decay and the competitive lateral inhibition from rival pathways, forces actively attempting to compress and close the channel. The brain resolves these competing vector forces mathematically in real time. This resolution defines the process of learning and decision-making. The macro-behavioral output of these equations dictates how an organism navigates its environment, locking the brain into a continuous cycle of habit, control, and flexibility. When a mathematically selected pathway is repeatedly successful, Hebbian plasticity physically deepens that specific strain channel. This forms a habit, a state of extreme efficiency that requires minimal executive control. The trigger for flexibility occurs when that deep habit encounters a prediction error or an environmental conflict. To break the established channel, the prefrontal cortex must exert heavy top-down override. This override pushes the brain into a flexibility state. By accessing a high degrees of freedom configuration, the network can test and reroute entirely new pathways before settling into a new habit. Brain evolution is the transition from isolated reflex loops to a globally integrated, phase-coordinated continuum. This fluid dynamic defines the architecture of advanced cognition.